search for genetic material. Even though the discovery of nuclein by Mr. and the proportion for the principles of inheritance by Mendel were almost at the same time, but that the DNA was the genetic material took a long to discover and to be proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes that are located on the nucleus of most of the cells. But the question of that molecule was actually the genetic material. There is a transforming principle that it was given by the Griffith. In 1928, Frederick Griffith has in a series of experiments with the Streptococcus pneumonia, which is a bacterium that causes responsible for the pneumonia. That is Streptococcus pneumoniae. During the course of this miraculous transformation in the bacteria, during experiment, the living organisms, that is bacteria, had changed in a physical form. When Streptococcus pneumonia, the bacteria that is grown on a culture plate, some produce smooth, shiny colonies. Let us take it as the S strain, while the others produce the rough colonies. Let us take it as R. This is because the S strain bacteria has a mucus, that is polysaccharide coat, while R strain does not. It has polysaccharide coat. Mice infected, let us see here, a strain that is injected into the mice, then the mice die because a strain, it is a virulent from pneumonia infection, but the mice infected with the R strain develop pneumonia. Inject into the mice, then the mice lived. The same way, Griffith was able to kill the bacteria by hitting them, that is a strain. Let us take here. Griffith was able to kill the bacteria by hitting them, that is S strain. It is heat killed. That is injected into the mice, then mice live. The same way, a strain, again heat killed, along with that R strain. It is injected. Here you can see it is heat killed, whereas mice lived, and R strain, that is in this case also the mice lived. It is injected, but the mice die. From this, he concluded that R strain bacteria had somehow transformed by the heat killed strain bacteria. So the transforming principle transfer from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was defined from these experiments. Here, that is biochemical characteristics of transforming principle. Prior to the work of Avery, MacLeod, and McCarthy, In 1933 to 44, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith experiment. They purified biochemicals, that is proteins, DNA and RNA, from the heat-killed S cells to see which ones transform the live R cells into the S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes, that is proteases, and RNA digesting enzymes, that is RNases, did not affect the transformation, and the transforming substance was not protein or RNA. Digestion with the DNases did inhibit the transformation, suggesting that DNA caused the transformation for this. They concluded that DNA is the hereditary material, but not all the biologists were convinced with this experiment. Later on, the genetic material that is given by the Hershey and the Chase experiments, and different experiments was taken, and they worked on the bacteriophages and viruses to confirm whether it is a protein or DNA is a genetic material. In next video, I'll tell about the Hershey and Chase experiment.